Welcome back. In the last session, we have studied the biosynthesis of penicillin, and we have seen how different structural variations can be done in the penicillin skeleton utilizing different tripeptides, where the valine is changed to other amino acids, and that can be converted into the different types of penicillin by the enzyme which is called isopenicillin N synthase IPNS. Okay. Now, let us talk about the other strategy where the gene which are producing the enzymes necessary for making the antibiotics, you manipulate at the genetic level and then get different types of antibiotics. Remember, I told you that there are two ways you can do these changes. One is by forcing the enzyme to accept alternate substrates. The other is by changing the genetic map, which will produce newer enzymes, and then you can get newer types of metabolites. Okay. Now, so we will talk about the second strategy now, and for that we will discuss we will discuss the what are called antibiotics, which are also classified as polyketides. Now, remember, polyketides basically represents a large group of secondary metabolites. What are secondary metabolites? Like penicillin, cephalosporin, vancomycin, all are secondary metabolites, not primary metabolites. What are primary metabolites? Primary metabolites is that without that you cannot survive. Secondary metabolites are basically elicited by for some other reason, like defense mechanism for the bacteria. So, they are secondary metabolites, like plants produce terpenes, plants produce other different natural products, they are called secondary metabolites, monoterpenes, sesquiterpenes. Now, there must be some reason why plant is making those secondary metabolites, but without that terpene the plant also survives. So, the primary metabolites are the ones which are required for growth, for sustaining the system, the living system. Secondary metabolites are elicited, are made just to augment their defense or attract, like it could be to attract the other partner, which are called pheromones. So, lot of things are there. Okay. So, they are secondary metabolites. Okay. Now, out of these secondary metabolites, one class is polyketides. What are polyketides? Polyketides are a large group of secondary metabolites which either contain alternating carbonyl groups, alternating carbonyl and methylene groups. That means COCH2. That means, if I write this CO, then CH2, then CO, then CH2, then CO, like this. So, this is a polyketide, if that is a metabolite. So, that will be a polyketide, or what it says are derived from precursors which contain such alternating groups. Like some compound may be there, secondary metabolite, which will be like this. Now, this is derived basically from this triketone. What happens after, in the process of generating the triketone, some of the ketone groups are reduced. Okay. So, this is also polyketide. Okay. So, basically polyketides are either alternating CH2CO, repeating units of CH2CO, or it could be units which are derived from repeating units of CH2CO. Polyketides, many of the polyketides have good antimicrobial properties, some are anti-cancer drugs and some are immunosuppressive agents. In fact, the major proportion of the natural products which are used as antimicrobial agents or anti-cancer agents are basically derived via polyketide mechanism, okay, or they belong to the class of polyketides. We will take one polyketide for our purpose, like in the earlier when we talk about the utilizing proteins to make new antibiotics, we took penicillin as our reference. Here we will take erythromycin. You know, erythromycin used to be a very good drug for upper respiratory treatment of upper respiratory infections. Today erythromycin has lost some of its most of its potency. So, this is the first generation. Then after erythromycin it came the roxithromycin, and then roxithromycin is replaced by azithromycin. Today azithromycin are prescribed. So, these are, but all are based on the skeleton of the erythromycin. Okay. Now, so we will take erythromycin as our example. Now, erythromycin is a macrocyclic compound. It is a big macrocycle that is present in erythromycin. Okay. I will show you the structure of erythromycin in some slide. It is there. This is not erythromycin. This is one of the precursor of erythromycin called 6-deoxyerythronolide B, but this is 6-DEB, 6-deoxyerythronolide B. Like isopenicillin N is the metabolite that is formed first, and then the that alpha amino adipoyl side chain is stripped off. So, you get 6 APA. Okay. So, the primary metabolite from penicillin biosynthesis is isopenicillin N, and the primary metabolite for erythromycin biosynthesis is this one. Then this was some other then post translational modification, then it is modified a little bit, put a sugar moiety that means form glycosides. Actually erythromycin is a glycoside. I think I have the structure somewhere in erythromycin. Yes, see, this is erythromycin, and you see the structure that I showed you is basically almost this one, except lacking the Wedge group. So, this is what is called 6-deoxyerythronolide. Let us again go quickly to that structure. Yes, you see only the Wedge is missing here. So, that is called the deoxyerythronolide B. Okay. Now, this is a class of, this belongs to the class of polyketide. Okay. Now, polyketide chemistry is or the biosynthesis of polyketides are basically repeating the same type of reactions over and over again, same types of reaction over and over again. Initially it may look quite difficult, but as the course progresses you will see this is not difficult at all, and you can always do retro biosynthesis. See, if I give a natural product, you can always break it down, and that is much easier for polyketide to break it down to the starting points, and you can then draw out how many enzymes are needed to make that polyketide. Okay. So, I will progress little bit slowly. We now know that polyketides are either this type of carbonyl system, or it could be hydroxy systems which is derived from the tricarbonyl, or even it could be a double bond here like this, because the Wedge can be or can undergo dehydration. So, basically if you see the structure, actually this is derived from these are polyketides, because the skeleton is derived from this tricarbonyl system. Okay. Now, let us take a very simple compound to start with. We start with suppose this compound 6-methyl salicylic acid, that is a natural product, 6-methyl salicylic acid it is one of the simplest polyketide, salicylic acid. Okay. Now, what is the structure? It is written here, but let me write again. There is a methyl, there is methyl, and then there is this, there is the methyl, there is this acid, and there is a Wedge here. This is 6-methyl. So, that is 1, 2, 3, 4, 5, 6 methyl salicylic acid. Okay. Now, this is apparently you might not be able to figure out that how this belongs to a polyketide or what is the mechanism by which this molecule is formed. Okay. Now, again I write that tricarbonyl compound in a different fashion. Suppose at some point of time this is made. Now, tricarbonyl compounds, if they cycle, see they can undergo aldol condensation. Suppose this carbon attacks here, forms a wedge and then the wedge dehydrates. Okay. So, if that happens, what you will get? A carbonyl here, here and here is a dehydration, because there is some reaction that has gone here. But this will now immediately tautomerize and form the, because the aromatic system is more stable. So, this will tautomerize and form this aromatic ring. So, that means the interesting point to note that when the tricarbonyl or tetracarbonyl whatever, when the polyketide chain is being made, it can undergo cyclization and form the aromatic rings. That is possible. And in many of the polyketides, you will see generation of aromatic ring or generation of very macrocycle compound like in erythromycin. Okay. Now, 6 methyl salicylic acid is one polyketide. I will tell you how you arrive at that. But apart from 6 methyl salicylic acid, you have another compound which is called orsinolic acid. I think this is where there is another wedge at the 4 position, another wedge at the 4 position. So, earlier this was 6 methyl salicylic acid, now orsilinic acid, maybe S, orsilinic acid. Okay. So, that compound, see that compound, if you try to figure out the position of the carbon oxygen bond, what you see, there is a carbon oxygen bond here, because this is CO2H, one carbon oxygen bond, and then there is a carbon, then there is another carbon which has got a carbon oxygen bond. Okay. So, that means you have basically a CO CH2 CO unit. CO unit was there, which was somehow transformed into the wedge, and then you proceed further, you have again a C and then another carbon oxygen. So, between all this carbon oxygen bond, there is a carbon which is present. Okay. Now, the question is, how do you know that there is a carbon oxygen here? That is little bit means that is you have to know that, or that you have to learn little bit extra. Means what I have said, that the polyketides are basically repeating units of carbon. This carbon oxygen could be in the form of carbonyl, could be in the form of wedge, or could be in the form of double bond also, because earlier I showed that it could be in the form of double bond also. Okay. So, double bonds are also suspicious functionalities, which maybe earlier could be a carbonyl, and then by cyclization chemistry it became a double bond. Okay. Like I showed here. Now, ultimately what happened, there is a wedge here, then a carbon, then another oxygen, carbon oxygen, then another carbon, and then now there is the question is how this ring formation has taken place. So, when there is a ring, you have to be careful that one of the carbon may be containing oxygen. Okay. Now, let us, because to make it little bit more clear, we have to proceed a little bit further. See, in a polyketide, what happens? You have already been told the formation of acetyl coenzyme A. which is a TPP catalyzed reaction, okay, pyruvate to acetyl coenzyme A. Now, you see the importance of acetyl coenzyme A. So, acetyl coenzyme A during the formation of polyketide, this is the starting point. One of the starting point is what? Acetyl coenzyme A. And to have the, because it contains more number of carbon atoms, so it needs another component, so that there is a reaction between the two. What is the other component? Usually the other component is CH2, CO, S, COA and C double bond OH. What is that? That is, this is called acetyl CoA and this is called malonyl CoA, because malonic acid only half of the only one of the carboxylic acid is converted into CoA. So, this is called malonyl CoA. Okay. Interestingly, this reaction generation of malonyl CoA is actually from acetyl CoA via a carboxylation reaction, and this requires biotin as a cofactor. Okay. I leave the mechanism of this reaction for the students who are attending this course, that biotin mediated carboxylation of acetyl CoA. So, this comes from the carbon dioxide, bicarbonate basically. Okay. Now, these two are the two units which react together and form CH3 CO, CH2 CO, S, CoA, that means plus carbon dioxide. So, what is the reaction here? That means this goes here first, that forms an anion here, and that anion reacts with this carbonyl that goes here, comes back, and this goes out. So, what it makes? CH3CO, CH2, CO, SCOA. Now, remember there is a reaction in organic chemistry called Claisen condensation, where you take ethyl acetate and add sodium ethoxide. What did you get? CH3CO ethyl acetoacetate. I think those who have studied organic chemistry, they remember it. Okay. So, what is the mechanism? That one molecule forms the anion and attacks the other molecule. Now, nature's way of making the anion here is not sodium ethoxide. Nature does not have sodium ethoxide. Okay. In fact, this is unstable compound. Any moisture will kill the sodium ethoxide, and these reactions after all is taking place in water medium. So, nature's way of making the anion is via decarboxylation. So, malonyl CoA undergoes decarboxylation and forms this what is called acetyl coenzyme A, okay, CH3CO, CH2CO, SCOA. Now, what happens? Usually what happens, that there is a reduction step, CH3, COH, H, CH2, CO, SCOA. So, its reduction, this is carried out by NADP or NADH mediated reduction, that we have already learned, that the reductions are taken place by NADH or NADPH. So, you get this alcohol, then you get a dehydration step minus H2O. So, that will form what? CH3 CH double bond CH CO SCOA. Okay. And then you get another reduction NAD, it could be NADH or NADPH, NADPH, and what you will get? You will get the saturation of the bond, of the double bond. Okay. This is exactly what is followed in case of fatty acid biosynthesis. In fatty acid biosynthesis, you start from acetyl coenzyme A, that undergoes Claisen condensation with malonyl coenzyme A, and then that gives acetyl coenzyme A, and then after these reductions, first NADH, then dehydration, then another reduction, So, you get CH3, CH2, CH2, CO, SCOA. So, this is a butyryl coenzyme A. So, you got the butyric acid. In fatty acid biosynthesis, what happens? This cycle continues, is repeated over and over again. Okay. So, then this one reacts with another malonyl CoA and you will get hexanoyl CoA finally, and then hexanoyl CoA goes to C8, then C8 goes to C10, C12, C14, C16. After C16, palmitic acid is C16. So, it falls off from the enzyme, the thioester goes off and becomes the acid. So, that is palmitic acid. Okay. Now, so there is great similarity between fatty acid biosynthesis as well as the polyketide biosynthesis, in the sense that here the first reaction is the same reaction that happens in polyketide also. However, in polyketide usually what happens, many of these, see actually this is the Claisen condensation, after that this is a reduction step. So, what is reduced? A ketocarbonyl. So, this enzyme, the enzyme which does that will be called Kr. What is Kr? Ketoreductase. Then this enzyme which causes dehydration will be called dehydratase, DH, and then the enzyme which does this reduction of the double bond is called ER. What is ER? Enoyl reductase. So, you have Kr, that is ketoreductase, you have DH, that is enoyl, that is dehydratase, and then you have ER, that is enoyl reductase. Why enoyl reductase? Because that functionality is an enoyl functionality, in and then the acid, enoyl. It is not acid like acetyl, okay, acetyl. So, this is an enoyl reductase. So, these enzymes are there. Now, in many of the polyketides, what happens? Because these steps will be used over and over again. So, when in polyketides, what happens? Some of these enzymes may not be present. See, if you want to make a really tricarbonyl compound, then these type of enzymes should be absent in the system, because you do not want any reduction of the carbonyl. If you want a compound which is say CH3, CO, CH2, CO, CH2, CO, and then CH2, CO2H. If you want to make a compound like this, then your enzyme system, what you are using, should lack all these ketoreductase, dehydratase and enoyl reductase, because they are only carbonyl, and carbonyl group is formed only in the first Claisen condensation. Okay. So, they have to be processed without touching those other three enzymes, ketoreductase. Okay. Now, let us talk about the first reaction, that is the Claisen condensation. Okay. How it happens? Basically, what happens? There is an enzyme called ketosynthase, which ends up as a thiol. So, this takes up the acetyl, see, ketosynthase has a thiol. So, ketosynthase SH plus acetyl coenzyme A, there is a thiol exchange now. So, the ketosynthase sulfur holds the acetyl group and coenzyme A leaves. Okay. And there is another enzyme protein which is called ACP, acyl carrier protein. What it has? It has got again an SH, ends up as an SH arm, and that takes up the malonyl CoA this is malonyl CoA. So, then ACP S CO CH2, then CO2H. So, basically before the Claisen condensation takes place, first this has to happen. A ketosynthase enzyme holds the acetyl group and an ACP, acyl carrier protein, holds the malonyl group. Okay. That means, what we are talking about is a ketosynthase here, which takes the S CO CH3 and an ACP side by side, and that has got S CO CH2 CO2H. Now, the reaction takes place, that undergoes decarboxylation. So, that attacks here, the ketosynthase is free. So, what will happen? The ACP is now holding the whole thing, the acetoacetyl CoA, CO, not CoA, ACP, acetoacetyl ACP, because CoA has already left. Okay. CO CH3, this is the compound. So, CH3CO, then CH2, then CO S ACP. Okay. Now, this S ACP, this compound, if there is a ketoreductase present, so this ACP is holding this whole 4 carbon chain, it shows it to ketoreductase. If it is present, then this carbonyl will be reduced. If there is a dehydratase enzyme, then it will show to the dehydratase. Okay. But all the time it is attached to the ACP. That means, you have several enzyme systems, ketoreductase or then dehydratase, then enoyl reductase. Suppose all are present, then what will happen? The ACP is holding this chain. So, there is a conformational change all the time. First, it shows if there is a ketoreductase, it puts the whole thing into the active site of the ketoreductase. So, that reduces the carbonyl. Then this is taken out, and then this is shown to the enoyl, the dehydratase, put into the active site of dehydratase. Then dehydration takes place, and then if there is enoyl reductase, it will put to the enoyl reductase. If all the 3 are present, then this CO will be saturated, it will be CH3, CH2, CH2, CO, S, ACP. Okay. So, now we know that after the Claisen condensation, and if all the that ketoreductase, enoyl dehydratase and everything is present, then we will end up with this butyryl coenzyme A. This was the CH2, which was earlier CO. Okay. Now, what happens? This has to be repeated. So, again a ketosynthase S, ketosynthase ACP, ketosynthase. Now, it has to be repeated. Okay. So, basically the reaction that takes place, you remember ACP always holds the malonyl group. Earlier in that reaction, ACP was holding the malonyl group and the ketosynthase was holding the acetyl group. Okay. In the next reaction, you again have another malonyl system. Now, COO, no, not COO, this is at C. So, that will be CO and then S. Now, we will talk about this, whether it is ACP or whether this is ketosynthase. We will talk about that little later, but a reaction again has to take place in order to extend the chain. So, like this. So, now you have extended the chain to 6 carbon CH3, CH2, CH2, CO, CH2, CO, S, and then whether it is ACP or ketosynthase. We will come to that point little later, but this is what is the chemistry that takes place. That means, now I can figure out that acetyl CoA has been used how many times to make this 6 carbon chain, acetyl CoA was used only one time, the first reaction, and malonyl CoA was used the first reaction, then the second reaction, then the third reaction and the fourth reaction like. So, acetyl CoA is called the starter unit and malonyl CoA is called the extension unit. Because your two carbons are added from the malonyl system. Okay. So, if that is clear, then we can slowly move into the earlier problem again, where we got little bit stuck. Okay. See, this was what we said, OSA or selenic acid. Okay. So, that compound, remember in the how many carbons are there? Let us count 1, 2, 3, 4, 5, 6, 7 and this one methyl 8 are there. Okay. Now, this I could see that there is a oxygen, carbon oxygen here, then a carbon, then there is this methyl. Okay. Let us again go back to that slide where I explained everything. See, at the end of the reaction, what you end up is a terminal CO2H, Okay. Like if it is palmitic acid, that means you have to have 7 cycles of this reaction. So, that 7 into malonyl unit means 2 carbon units, that is 14 plus acetyl CoA, that is 2, that means C16 is generated, and then if it falls off from the enzyme system, you will get an end is the CO2H. Okay. Now, if I say that how many malonyl units are present here, so what you do, you start from this CO2H. So, this is one malonyl unit, then this is another malonyl unit and this is your starter unit. All cannot be malonyl unit, then where is the starter unit? Or you can start from this side, first 2 carbons is your starter unit, then the second 2 pair of carbons is your coming from the extension unit. So, that is malonyl, this is also malonyl. So, if I have more carbons now, 1, 2, 3, 4, 5, 6, 7, 8. So, it is suppose CO2H. So, this is one malonyl unit, that is one malonyl, that is one malonyl and this is the starter unit. Okay. So, that means malonyl CoA has reacted 3 times here. Okay. So, again we go back quickly to that compound. This compound I want to dissect it into a polyketide. See, this is I can start from the end point. See, the CO2H is here. Okay. Then there is a carbon, then CO2H, then there is a carbon, then I go this way because there is oxygen here. I know that the 2 oxygens here, this one and that one is 1:3 relationship. So, I do not go that way, what I do is CO2H, then the carbon, then there is this carbon which must be having a carbonyl or a derivative of the carbonyl, then a CH2, then another carbonyl, then CH2, then another carbonyl and then methyl. So, if you dissect it, then you should obtain a system like this: CO2H, CH2, then CO, then a CH2, then CO, then a CH2, then a CO. So, now what is the starting point? The starter unit is acetyl coenzyme A. Then there are how many malonyl units here? What is the extension unit? A malonyl. So, how many malonyl units are there? This is one, this is another and this is another. That means, there are 3 cycles of reaction that has taken place, that Claisen condensation, followed by other enzymatic reactions that has taken place. Okay. So, what was found that in this if you go by that there are 3 Claisen condensations have taken place. I think this is not the correct picture. What I will do, I will again go back to a blank slide. See, usually this is the picture of the protein. This polyketide can be synthesized, or that is, it has been found that there are two or basically 3 types of polyketides, but we will concentrate on only 2 types. One is called type 1. What is type 1? Type 1 is that you have a big enzyme, big protein, not enzyme, big protein, and then in the big protein you have domains of different activities. In a single protein, you have domains of different activities. Like suppose this is a ketosynthase, it has got a ketosynthase activity. This is got, suppose this is keto reductase, they say. Suppose this is a dehydratase. I will talk about this, what is AT, I will come back, AT and then ACP. Okay. So, you have basically this is what is called type 1 polyketide, type 1. Type 1 is what? You have a big protein where different domains of activities are there. Okay. Different domains of activities are there. And type 2 is you have different proteins, ketosynthase, ketoreductase, all these, and at the time of synthesis, biosynthesis, they all come together and then have a cooperative effect between them. So, first one reaction takes place here, that transfers it to here, then that transfers it to here, that transfers in a cyclic manner. Okay. So, this is type 2. So, type 2 is different proteins assembled together at the time of biosynthesis. Type 1 is a single protein containing different functionalities at different domains. Okay. So, now let me see which way we should go. The first reaction I told it is basically between a ketosynthase holding an acetyl group and an ACP which is holding the malonyl group. That is the first reaction. Okay. Now, the question is who is putting this acetyl group to the ketosynthase? Is it that is also done by another enzyme? See, my hand is here, but I now left hand is attached to an acetyl group, but some enzyme has to come and then bring it alongside and then put it on my left hand, and on the right hand suppose this is ACP. So, again the same problem here, this right hand is just sitting here. So, another enzyme will bring. So, there must be some carrier protein which brings the starter and the extension unit and attach it to ketosynthase and ACP. Okay. I hope that is clear. So, that means all these polyketide synthase in the type 1, we are talking about type 1 cases, we are not talking about this separate enzymes, one protein having all the activities. So, it starts with like this, it starts with what is called an acyl transfer. This is nothing but an acyl transfer. You are transferring the acetyl group from acetyl coenzyme A to the ketosynthase. You are transferring the CO is CoA. You are transferring the malonyl group, that is also an acyl group, and then putting it to the ACP. So, this is what is called AT. AT's job is to put the do this transfer. Acetyl group transferred to the ketosynthase, malonyl group transferred to the ACP. So, AT is acyl transferase. Okay. So, initially there is an acyl transferase here, and then so all polyketide synthase starts with this acyl transferase, because that takes up the acetyl group, acetyl coenzyme A, and there is an ACP also here. So, first this AT puts the acetyl group onto the ACP. ACP, remember, has an SH. So, first acetyl transfer takes place to the ACP. Who does that? The AT. Now, this is what is called the loading module, because this is like a primer kind of thing. See, loading module is that where you start the whole thing. So, the acyl transferase first takes the acetyl coenzyme A, gives it to the ACP, okay, by a transthioylation reaction. Okay. So, acetyl group is now in the ACP. Next, the ketosynthase has SH. So, what it will do? It will, now the SH is holding the COCH3. Now, the ketosynthase will, this acetyl group which was at the ACP, that will be transferred to the ketosynthase. Okay. So, again I repeat, because this is little bit complicated, but once you know this, that becomes little, that will become easy, I am sure. So, what happens? There is a loading module. The AT transfers the acetyl group to the ACP. ACP also belongs to the loading module. Now, the ACP transfers the acetyl group to the ketosynthase. So, basically it is a two-step process. Directly it does not go to the ketosynthase. It was first given to the ACP, ACP shows it to the ketosynthase, and ketosynthase SH. Remember, ACP has SH and ketosynthase has SH. Okay. So, this is nothing but this ACP first holds the acetyl group and then shows it to the ketosynthase. Now, ketosynthase attacks this acetyl and frees the ACP, and thereby it is holding the acetyl group. So, first ACP, which is done by AT, and then ACP to ketosynthase. So, it ends there. The loading, your starter unit is now being held by ketosynthase. Now, there is another acyl transferase here. What it does? It puts the malonyl group into this ACP. Remember, this ACP is different from that. This belongs to the loading module, and this is what is called the first module. Means there may be several modules like this. This is first modules. So, ketosynthase has the acetyl group, ACP has the malonyl group. Who transfers the acetyl group to the ketosynthase? It is the acyl transferase in the loading module from the ACP which does this. Okay. So, now there will be reaction between these two, that Claisen condensation. So, what will happen? ACP will hold the SCOCH to COCH3 after the reaction. Now, because there is a ketoreductase, it will show the whole chain to the ketoreductase. So, ketoreductase now reduces the carbonyl. There is a dehydratase. So, the dehydration will take place, but there is no enoyl reductase. If that is not there, that means after this reaction you get the double bond compound. Okay. Now, what will happen? All the time this is being held in the ACP. So, always there is the second module, when it is the next cycle of reactions take place, there is a second module. It is not repeated, that this is again thrown back to the ketosynthase, not like that. It is a modular process. So, first module has this type of framework, this type of domains. So, once that is done, then it is transferred to the ketosynthase of the next module. Okay. And the next module suppose has only ketosynthase, acyl transferase and ACP. Then what will happen? If I ask what is the product at this end, then what will be the product? I can write it CH3, then CH double bond CH, then CH double bond CH, then CO, then CH2, then CO ACP, SACP basically. How do I know this? Because the starting in the, see, this is the our starter unit. Now, in the first module what you have is every module should have ketosynthase and acyl transferase and ACP, that is mandatory. In the first module I see that there is a ketoreductase and dehydratase, that means after the first reaction a double bond is formed. In the second module I do not see any 1, 2, 3, 4, 5, 6. In the second module I do not see any ketoreductase or dehydratase or anything. So, that ends up as the carbonyl and then CH2S, CO S ACP. Okay. So, basically then we will end up by showing this. This is the deoxy erythronolide B. Okay. Now, what is my starting point? The starting point, as I said, the end point is a carboxy and the starting point is either acetyl. It could be acetyl, but do not take it sacrosanct that always the starting unit is acetyl. It could be propionyl, it could be butyryl, all these are possible. Similarly, the extension unit can be malonyl or it could be methyl malonyl. There are different types of extension units are possible. Okay. Now, this 6 deoxyerythronolide, I see there is a lactone moiety. First check whether where is that acid group, because it has to end at the acid. So, I see that there is a lactone. If you hydrolyze this, what will happen? If you hydrolyze this, that will become OH and this becomes CO2H. Now, you work backwards. So, this is your one extension unit, that is your another extension unit, this is your third extension unit. Actually, we are going in the reverse direction. This is the fourth extension unit, this is the fifth extension unit, this is the sixth extension unit. Okay. So, there are 6 extension units and now this is your starter unit. Okay. Let me write the structure again. This is very written in I again write the structure. So, here there is this bond what I actually deleted, because there is a carbonyl here. So, I deleted sorry. So, what happens? Maybe I put the OH here now and CO2H here. Okay. Put there is a ethyl, forget about the stereochemistry, there is a ethyl here, there is methyl, there is after every CH there is a methyl, then there is a methyl here, then there is a methyl here, then there is a methyl here. Okay. Let us see where are the oxygens. One oxygen is here, OH, this is a carbonyl, this is this is nothing, this is CH2, then there is a OH here. Again I repeat, there is, we are not thinking about the stereochemistry here, and then there is a OH here. Okay. Now, I can write what are the enzyme components in different modules. First of all, how many modules it should have, that is number 1, apart from the loading module is separate. There should be 6 modules, because 6 extension units are here. What are those 6 extension units? One is this one, this is your starter unit, this is the extension unit, maybe I use another color. This is your extension unit, this is your extension unit, this is your extension, this is the extension, this is the extension, and this is the final extension unit. So, 6 extension units, that means you have to do 6 Claisen condensation. So, there are 6 modules in this. Again I repeat, apart from the loading module. So, it started all from here, this is the starter. So, what is the starter unit? It is not acetyl CoA, it is propionyl CoA, because you have an ethyl group here. So, this is your starter unit, and what is your extension unit? It is not malonyl CoA, but it is a methyl malonyl CoA, SCOA, there is a methyl group and then CO2 each, because everywhere you have a methyl extra. If this methyl was not there, then malonyl CoA would be the compound, but it is methyl malonyl CoA. So, now let us see, the first Claisen condensation has put a wedge group here, but no dehydration, no saturation. So, the first module I can say what will be there? Ketosynthase, acyl transferase and ACP are mandatory. Then we have to see what is present, ketoreductase, dehydratase or enol reductase, because it lands up in wedge. So, there is a ketoreductase, you see. It does not matter what is the order of this, you can write anything. Order is not important here, we are trying to understand. So, ketosynthase, acyl transferase has to be there, ACP has to be there, and then ketoreductase. So, it ends up with OH only. Second one again it ends up with OH. So, the second module again same, ketosynthase, acyl transferase, ketoreductase, ACP. Third module it ends up at a carbonyl. So, that means there is no ketoreductase also. So, third module ketosynthase, acyl transferase, ACP. Fourth module I see there is CH2, no carbonyl. That means all the domain your domain activities are present. That means it will have ketosynthase, acyl transferase, dehydratase, enol reductase, ketoreductase and ACP. Again I repeat, you can have any order. The most important is you should identify that domain activities in each module. Then the module, so this is the fourth module, then fifth module you end up as the wedge. That means it should have a ketoreductase, ketosynthase, acyl transferase, ketoreductase, ACP. And final, this is the other one, the last module has what? Last module again ends with OH. So, it will have ketosynthase, acyl transferase, ketoreductase, ACP. Now, the last one you have to release it. How do you release it? That is called a thioesterase, because after all your ACP is what is holding, na? S, CO and then the whole chain. So, now you have to have an esterase enzyme, which, but that is the thioesterase enzyme, which hydrolyzes this one. Okay. So, that is the release always thioesterase and loading always AT and ACP. See, now it is more or less understandable, I believe, that if you can identify all these where are the oxygens and then try to find out the starter and the extension unit, you can devise, you can tell what are the modules. If I say that this is a type 1 polyketide synthase. Okay. So, next day what we will do, next session, we will start from here. See, now we have these modules and there are scientists, like there is one Indian scientist, his name is Chetan Khosla, and he has many YouTube videos about polyketide synthase, especially erythromycin biosynthesis. What he has done, he has, suppose I take this thioesterase domain and put it after module 2, then what will happen? Module 1 reactions will take place, module 2 reactions will take place, then the molecule will be released. And then I can get a new type of compound, because sometimes this thioesterase it can actually form macrolactonization. Means, like in this case in erythromycin, what happens, this wedge, last wedge actually is attacking this carbonyl and releasing the sulfur, but this is an enzyme catalyzed reaction. So, it is a macrocyclization, one wedge and one carboxy, they reacted with each other forming the macrocycle. So, that is the biosynthesis of erythromycin. So, now you see you have the genetic map. So, you can cut off different domains. Suppose I want an erythromycin where it is not carbonyl, it is only CH2. So, what I have to do? I have to add domains. I have to add a keto reductase, a dehydratase and enoyl reductase, then I will get a CH2 here. Okay. So, now this is the trend of today, that how do you manipulate the gene, especially after this is known that these are modular enzymes, okay, and then you can make new types of secondary metabolites, and which some of them may be very useful. In fact, erythromycin, different types of truncated erythromycin have been prepared by the same person, Chetan Khosla. He is a scientist at Stanford University. Okay. So, we will work out some problems in our next session. Thank you.